Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Victoria Rees, editor of Drug Target Review. Today's speaker will be Dr. Jean Bernatchez, senior scientist and San Diego R&D group leader at Eurofins Discovery. Following his presentation, we'll be moving on to a live question and answer session where you can pose any questions that you have to today's speaker. Please remember that you can submit your questions at any point during the webinar using the questions panel, which is situated in the menu on the right. But without any further hesitation, may I pass over to Jean?
Thank you for the kind introduction. Welcome everyone, and thank you for choosing to attend this webinar on Eurofins Discovery's new STAT SH2 domain binding assay platform for the identification of small molecules that selectively bind to STAT family proteins. In today's presentation, I will be discussing STAT transcription factors as a drug target, as well as presenting an overview of Eurofins Discovery's portfolio of binding assays for small molecule drug discovery. I will show validation data for our soon-to-be available STAT binding assays, as well as how the STAT panel can be coupled to our E3-scan platform to develop bifunctional protein degraders of STAT proteins. I will end with an overview of our targeted protein degradation portfolio and then take questions at the end of the presentation. Through our drug discovery experts, Eurofins Discovery can assist at the various stages of your drug discovery projects, providing services and products in the areas of synthetic chemistry, in vitro pharmacology, safety pharmacology, custom assays and products, in vivo services, and translational phenotypic services. While the scope of today's presentation will focus on our in vitro profiling assays, be sure to reach out to our service representatives if you're interested in any of our other offerings for your drug development projects. STAT proteins play critical roles in canonical signaling pathways in cell growth and differentiation, and in inflammation. In addition, functions related to gene repression, as well as roles for unphosphorylated STAT proteins, have been identified. Deregulation of the biochemical functions of the STAT proteins have been associated with cancer and autoimmunity. This has driven researchers to develop small molecules targeting this class of proteins. Eurofins Discovery has developed a new panel of STAT SH2 domain binding assays to assess the affinity and selectivity of small molecules targeting the SH2 domain of STAT proteins. This technology builds on our well-established binding assay technology platform, which has been used to generate binding assays for numerous protein families, including kinases, bromodomains, BCL2 proteins, and E3 ligases. In a STAT SH2 domain binding assay, three key components are present: the SH2 domain of a STAT protein tagged with DNA, a known ligand of this SH2 domain immobilized on a solid support, and a test compound or solvent control. The DNA tagged SH2 domain of interest is incubated with a known ligand immobilized to the solid support. In the presence of a competitor test compound, the target protein is competed off the solid support. After washing of the beads and dilution of residual protein bound to the solid support, the amount of DNA tagged protein is quantified via an ultrasensitive qPCR readout. In the presence of a competitor test compound, the final qPCR readout is expected to have a low signal. In the presence of a solvent control, very little protein is competed off the solid support. After washing of the beads, elution of residual protein bound to the solid support, and qPCR quantification of the DNA tagged protein, the assay signal is expected to remain high. A dose response of a competitor compound can be tested using this assay, and an accurate thermodynamic dissociation constant for the competitor can be extracted from the resulting data. SH2 domains in the STAT binding assay platform are expressed in whole mammalian cell lysates as a fusion construct with the NF-kappa B DNA binding domain. An exogenous DNA probe is added to the SH2 domain containing lysate, which anneals to the NF-kappa B DNA binding domain. The DNA probe also contains the qPCR amplicon, which allows for detection of the protein of interest in the assay readout. The STAT panel offers 7 wild-type STAT SH2 domain binding assays, as well as 2 STAT3 mutant SH2 domain binding assays, and 1 STAT5B mutant SH2 domain binding assay. To validate the STAT panel, we utilize compounds selected from the scientific literature, which have been shown to be selective for their specific targets. The STAT3 SH2 domain targeting small molecule SI109 and its related bifunctional protein degrader SD36 were profiled against the STAT panel. In addition, the STAT5B SH2 domain inhibitor STAFIB1 was profiled in the STAT assays. Sample curves for the binding of SI109 to STAT1, STAT3, and STAT6 are shown on the left, while selectivity data for the 3 compounds used to validate our panel are shown in the table on the right. Rank order and selectivity of the compounds tested tracked closely to the affinity values published in the scientific literature. The exquisite sensitivity of the qPCR readout of the assay system allows for the use of low picomolar amounts of protein in our assays. As such, our assays do not approach the tight binding limit even for compounds with picomolar affinity, allowing for the measurement of a broad dynamic range of KDs during compound profiling. Furthermore, during assay development, the bait loading of the immobilized capture ligand on the solid support is optimized so as to not have an effect on the measured KD of the competitor. As such, true thermodynamic KD values are reported for control and test compound competitors. In addition to off-the-shelf assays available in our screening panels, we also offer custom assay development services on a case-by-case basis. Here is a flowchart for the general workflow for a custom assay development project. The initial stages, which involve cloning and synthesis of the immobilized affinity ligand and the control competitor, as well as the assay proof of concept and optimization, take generally 6 to 8 weeks, while compound screening, hit confirmation, and potency determination is performed in the 2 to 3 weeks following assay onboarding. There are major advantages in using the STAT binding assay platform for drug discovery programs. Collected data are accurate, precise, and reproducible, and there is a high sensitivity and a broad dynamic range in KD measurements for competitor compounds. We have internal reference compounds that we have characterized for binding affinity, and all assays are run in parallel on a single platform, allowing for direct comparison of data between targets. This is the largest assay panel available on a single technology platform for STAT protein SH2 domains, all without having issues of compound interference in the assay readout. Assays are performed in a high throughput 384 well format, allowing for library profiling. We also offer custom assay development on a case-by-case basis for your protein target or mutant of interest. We offer rapid turnaround for compound screening of 5 days for weekly submissions and 20 days for library screens. While major advances in traditional drug discovery have allowed for the inhibition of important disease-associated targets, 80% of human proteins have largely remained out of reach for therapeutic design due to their lack of a catalytic or regulatory site. The emerging field of targeted protein degradation, or TPD, has allowed for the targeting of proteins that were previously thought to be undruggable. STAT proteins are prime candidates for the development of bifunctional protein degraders through the design of selective ligands for their SH2 domains. The principle of TPD relies on the cellular machinery responsible for the natural cycle of protein turnover of the cell, described by the ubiquitination proteasome pathway. In this system, the E1 enzyme activates ubiquitin, which is then transferred to the E2 enzyme. Ubiquitin is transferred to a target protein slated for degradation via the E3 enzyme. Polyubiquitination of the target protein marks it for degradation at the 26S proteasome. Ubiquitin is then recycled back into the pathway. In targeted protein degradation, a small molecule can be developed which hijacks an E3 ligase, directs it to polyubiquitinate a novel substrate protein through induced proximity, which then marks the novel target for degradation at the proteasome. Two popular strategies for TPD are shown on the right: bifunctional degraders and molecular glues. In the case of bifunctional degraders, one warhead specific to a substrate recognition domain of the E3 ligase is connected via a chemical linker to a warhead which can bind to a protein-protein interaction domain, allosteric site, or catalytic site of a target protein of interest. In the case of molecular glues, the small molecule can induce a conformational change in either the E3 ligase, target protein, or both proteins that reveals a surface where both proteins can bind together, facilitating ubiquitination of the target protein through induced proximity. Eurofins Discovery has developed a panel of E3 ligase binding assays, known as E3-scan, to assist with the discovery of novel E3 ligands for the design of targeted protein degraders. This panel can be used in parallel with our other binding assay platforms, such as KinomeScan, BromoScan, and BCL2-scan, to identify ligands for target proteins of interest designed by functional degraders, cell-based and phenotypic assays to evaluate the functional effects of designed protein degraders on the level of a target protein of interest, can be performed using Eurofins Discovery Sprinter, InCell, and Biomap platforms, and our synthetic chemistry services can assist in crafting the right protein degrader for your project. Eurofins Discovery now offers 26 E3 ligase binding assays spanning 19 E3 ligase targets. The E3-scan platform is available in two formats. First, we offer ScanElect, where test compounds are screened at a single concentration, and compound activity is provided as a % of an internal control. There is no curve fitting at this point, and a minimum 100 compounds is required for submission. The second format is KD-Elect, where compounds are screened in 11-point dose response in duplicate, and the data is provided as curves, including KD binding affinities. The E3-scan panel can be used in parallel with our SH2 STAT protein binding assays to identify ligands for both an E3 ligase of interest and a STAT SH2 domain of interest during the design of a bifunctional protein degrader. In summary, I presented an overview of the new STAT SH2 domain binding assay platform offered by Eurofins Discovery, which can be leveraged for the discovery of selective small molecule inhibitors targeting the SH2 domains of STAT proteins. We provide validation data for this panel using small molecules, and a bifunctional degrader from the scientific literature, and recapitulated published rank order and selectivity data for these compounds. The STAT assay platform can be used in parallel with our E3 scan platform for the design of bifunctional degraders targeting STAT proteins. I presented an overview of the technology platforms offered by Eurofins Discovery in the targeted protein degradation space, with an emphasis on the expanding E3 scan biochemical binding assay platform. Profiling compounds on the E3 scan platform can be coupled with compound screens on our kinome scan, bromoscan, BCL2 scan, and STAT SH2 domain platforms for the rational design and development of bifunctional protein degraders. With that, I would like to thank you all for attending, and I would be happy to take questions at this time.
Fantastic. Thank you, Jean, for that excellent presentation there. So we'll shortly be moving on to our question and answer session, where Jean will answer all of your questions. Don't forget that you can submit any questions that you have using the questions panel, which is in the menu on the right. But before we get into the questions, we have some quick polls for you. So our first poll for you is, are you currently interested in assays or panels for any of the STAT proteins? So all you have to do is just select either yes or no. Fantastic, thank you. So as we can see there, we have 33% who selected yes and 67% who selected no. And here's our next poll question for you. Are you interested in developing inhibitors or bifunctional degraders of the SH2 domain proteins? So again, all you have to do is just select either yes or no here. Fantastic, thank you so much again. So as we can see here, the results of this one are 42% of you selected yes and 58% of you selected no. And this quick poll question, what service areas would you like to see more information about? So for this one, you can select all that apply, and our options are STAT proteins, E3 ligases, kinases, GPCRs, and safety pharmacology. So reminder, you can select all that apply here. Brilliant, thank you so much for all of your answers there. So we are going to move on to our Q&A section now. So again, you can still send in your questions using the questions panel, which is in the menu on the right. So let's get into it. Our first question for you today, John. Will you develop a custom assay that is not in your current menu and terms?
Thank you very much, Victoria. Yes, we have the capacity to develop assays that are currently not in our off-the-shelf assay offerings. Usually this involves a discussion with the client about the potential viability of the project based on existing literature, particularly with respect to the cloning of the target of interest and existing data on the capture ligands that are present in the literature. If the project is deemed feasible, then we will engage in a custom assay development project with the client, with the understanding that there will be screening performed at the end of the custom assay development project. And that varies based on the projects we receive. So we'd be happy to entertain any inquiries as to custom assay development projects. We perform these on a regular basis.
Fantastic, thank you. So next question for you. When will the stat panel be ready for compound testing?
It will be ready in the next couple of months. We're anticipating this to be offered soon. If you're interested in this particular panel, please reach out to us and we can make sure that we can get started on your project as soon as possible.
Fantastic, thanks, John. So can this assay detect compounds that bind to another domain besides the SH2 domain on the stat protein?
In this particular case, it's unlikely, given that we actually clone specifically the SH2 domains rather than the full-length stat proteins. However, in certain cases, you might be able to detect compounds that stabilize a conformation of the protein that is itself competitive with the binding of the capture ligand, and so that would be detected. But in most cases, you're going to find compounds that directly compete with the or overlap in terms of their binding with the binding pocket for the ligand, the capture ligand that's on the salt support.
Brilliant, thank you, John. The next question for you. For this assay, does the KD of the test compound, is the KD of the test compound dependent on the affinity of the capture ligand to its stat target?
That's a good question. For every single assay project that we initiate, we always perform a bait load gradient where we empirically determine the bait loading at which the concentration of the capture ligand on the bait load no longer influences the KD of the competitor test compounds. So this is done empirically for every single assay. And so to answer the question, no, the bait loading does not have an influence on any of the KDs of the capture of the competitors tested in our assays.
Fantastic, thanks, John. Are there examples of small molecules tested in stats and the E3 ligase?
Sorry, can I have that question again? Sorry.
Of course. Are there examples of small molecules tested in stats and the E3 ligase?
Yes, so I presented a few today that were published in the scientific literature. SI109 and ST36s are both commercially available small molecules. There are a number that are currently in clinical trials and have not been disclosed yet, but there are other molecules that we've tested internally in-house. I'm only showing a certain selection of the data today, but yes, there are additional ones that are commercially available and that are also reported in the literature that can be synthesized.
Brilliant, thank you. And just a reminder that we do still have some time, so if you have got any questions that you'd like Jean to answer, still time to send them in. So Jean, next question for you. Do you offer assays as a panel or individual assays for stats or the E3 ligase?
So we're going to be offering it both as a full panel and also as an ad hoc selection of assays, depending on what the customer's needs are. If you want to just screen against a certain number of these assays, that is totally fine as well. We also offered it as the full panel as well.
Lovely, thank you. So does this assay also apply to the undruggable phosphatase family proteins such as SHP2?
We can look into any undruggables or classically undruggable targets that you're looking at. The most important aspect for a custom assay development project to take off is really the presence of literature on capture ligands, preferably reported dissociation constants for capture ligands, be they either peptides or small molecules. These are things that I always look for to assess feasibility and viability of a specific project in that space. But we have done this before in the past. We've worked on a number of undruggable targets. You know, a lot of the popular ones, scaffolding proteins. You know, as you mentioned, phosphatases are definitely possible as well. So that's what.
Lovely, thank you, John. And just another quick reminder, we've had some fantastic questions come through today, so please do send them in. John, next question for you. Do you offer any cell-based assays for protein degradation?
Yes, I didn't get a chance to mention it very much in the talk today because it was focused mostly on biochemical assays, but in one of my slides I had also our in-cell and sprinter platforms that are available to detect degradation of target proteins of interest. And so if you're interested in this particular component, please reach out to our customer service representatives and we can put you in touch with our subject matter experts in this particular area. So yes, we do have cell-based targeted protein degradation assays.
Brilliant, thank you. And what is the turnaround time for custom assay development for this assay?
So generally speaking, as I mentioned in the slide deck, it's between 6 and 8 weeks. If it's for a mutant, then if it's a simple point mutation, then it probably might be a little shorter. But for a full clone, you know, design and ordering of the clone and expression of that particular clone, and then optimization of the assay, it's usually around 6 to 8 weeks. Then there's approximately, you know, a couple of weeks to a month onboarding of the assay into production, and then following that, production has its own timelines, as I mentioned in the talk, 2 to 3 weeks for library turnaround time.
Lovely, thank you so much, John. Fantastic, thank you for all of those questions that were sent through. Thank you, Jean, for answering all of them. So please do join me in thanking Jean for his time today. I think we've answered all of the audience questions that have come through, so thank you once again, Jean. As you leave the webinar, a survey will appear on your screen asking you to rate this webinar. Please do take a moment to provide your feedback, and if now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. But now, on behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar, and thank you once again to Jean. We hope to see you again soon. Goodbye.